So today we are going to discuss about the CRISPR, which is a genome editing tool. In the genome editing tool, you can edit the gene. And while doing the editing of the gene, how you can do the editing of the gene? Either by the knocking out the gene, or knocking in the gene. In the knockout gene, you are going to delete certain sequence of the gene so that the function of that gene is going to be disrupted, or either you can delete the entire sequence. In the knock-in mutation, you are going to correct the gene. For example, in what happened in certain diseases, the disease is caused due to the error in a single amino acid. So you can correct that amino acid with the correct amino acid. So this is the knock-in mutation that happened in the cystic fibrosis and certain other diseases as well. So nowadays this technology is in high demand. Let's focus on its full form. What is the full form of the CRISPR? As you can see here, the full form of the CRISPR is the clustered regularly interspaced short palindromic repeats. So what are the palindromic repeats? So the palindromic repeats are those repeats if you are reading them from left to right or right to left, they are looking simple. For example, if you are see this sequence NGGN. If you are reading them from left to right, it is NGGN, and if you are reading from right to left, it is again NGGN. So these types of sequences are known as the palindromic sequences. And what happened after these palindromic sequences? There is a spacer molecule, and then again there will be N, G. I don't know what happened to this one. G, N, N. Span is not working. Let's see here. So this is the palindromic repeat. This is the spacer molecule, and this is again the palindromic repeats. So you can see the short palindromic repeats, they contain the spaces in between and they are clustered regularly, and that is why the full form of this technology is CRISPR, is clustered regularly interspaced short palindromic repeats. Okay. This is a kind of immune system in the bacteria, and the virus is virus is infecting the bacteria, while bacteria is showing the immune response, and that immune response is in the form of CRISPR. For example, if virus is attacking the human body is showing the specific or the non-specific type of immunity. Similarly, the bacteria is showing the immune response against virus, so that the same virus cannot attack the bacteria one more time. So as I said, this is a new gene editing technology. But before that, there were other techniques as well. For example, there was talons and the zinc finger techniques for the gene editing, in which you can either do the knockout mutation or you can do the knock-in mutation. So what happened in the zinc finger nucleases and the talons? In the talons specifically, there are 32 to 33 amino acids which are conserved in region, which are very much conserved. Only 2 amino acids are different, and they recognize the sequence of the DNA which you want to target. So what happened in this talons, the protein, because amino acid is part of amino acids from the protein. The protein is guiding this FOC1 restriction endonuclease, where this endonuclease should cut the DNA. So it is a protein guided. Here the protein is guiding the restriction endonucleases, where it should cut the DNA or chop out the DNA. Similarly, in the zinc finger nucleases as well, this is the protein that is guiding the endonucleases, where it should cut the DNA. So both of these are protein guided. In case of the CRISPR, here the RNA is guiding the Cas9 endonuclease, where it should cut the DNA. This is the RNA guided phenomenon. So here you can see ease of design is pretty much simple in case of the CRISPR Cas9 in comparison to ZFN and the talents. As I said, there are 32 to 33 amino acids and 2 amino acids that will vary and that will recognize the sequence which you want to target. Okay. So and there are very less number of softwares are available. If you are going to perform your gene editing experiments by ZFN and the talents, you have to use those rare softwares and design the experiment. And let's say if your experiment doesn't work, then you have to redo the experiment. But what happened in the CRISPR-Cas9, you can design multiple sgRNAs and can do the experiment in a day or two. It's very simple to do this experiment in comparison to the talents and the ZFM, because you have to design a long sequence for targeting your gene of interest. Okay. Now you can see this is the off-target effects. And what is the off-target effects? As you can see here, here is the sequence A, double G, and C. Let's say if you want to target this sequence, and we want only this sequence to be targeted, not the other portion. So what will happen? In case of the CRISPR-Cas9, there are chances the Cas9 will recognize instead of that will recognize this sequence, as well as there are chances that the same sequence could present somewhere else as well. So this Cas9 may cut that portion also. So this is known as the off-target effects. Our target is this, but we are creating a off from this target. So the other portion could also be chopped out. So there are chances of the off-target effects in the CRISPR-Cas9, but in case of the ZFN and the talents, there are very less chances. And why the chances are less? Because you have to design a long sequence, and these restriction endonuclease will form the dimer. And because of this, chances of the off-target effects are pretty much less. But in the CRISPR-Cas9 also, nowadays scientists have created lots of mutation in this Cas9 protein so that the off-target effects are less. Okay. So what is the CRISPR-Cas9 system if you think about it? So as I told you already, these are the palindromic repeats. Palindromic repeats are those repeats which you are reading. If you are reading them from left to right or right to left, they are looking similar. There is a spacer molecule in between them, and near to these spacer and the repeats, there are the Cas genes. And what are these Cas genes? Cas genes are Cas1 and the Cas2. So if the virus genome is DNA, that the Cas9 will work, Cas1 will work. If the virus DNA, if the virus genetic material is RNA, then the Cas2 will work. And both of these enzymes have two activities: integrase and the nuclease. So what is the function of nuclease? When the virus will attack the bacteria, this nuclease will chop out that or cut the DNA and integrate the DNA in between these repeats. So the spacers are going to be replaced by the viral genetic material, whether it is RNA or DNA. So this is the CRISPR locus. So now let's say if this is the viral DNA is there, and then once the sequence is transcribed in the bacteria, it will convert into the RNA. So next time the virus will attack, what will happen? The DNA and the RNA, they are complementary. This is complementary sequence, so they will bind to each other, and in this way, this is the guide RNA that will guide the Cas9 to chop out the viral DNA. So to this bacterial, this portion, this is the guide RNA that will guide the Cas9 to chop out the genetic material of the virus, of the same virus. Okay. So we will describe it in detail. So let's talk about something about the type 1 and type 2, 3 and 4 types of CRISPR. Type 1 and 3, they are using multiple Cas proteins, and type 2 and type 4, they are using a single component Cas9 protein. Type 2 is the most common. As I said, this is a single component Cas9. So there are many vendors which who are having the vectors that is expressing the Cas9, so you can just go and purchase it. While I don't think so the vendors are having, or the work has been focused on the type 1 and the type 2, because there are multiple Cas proteins. It's difficult to assemble them in a single vector. So here we will understand about how the CRISPR type 1 system will work. Let's say this is the virus, this is the bacteriophage virus, this is the viral genetic material. It is attacking the bacteria. Then because of these Cas genes, as I said, these Cas genes have the integrase in the nucleus activity that will cut the DNA, and that DNA is going to be integrated in between these repeats. Now, the while this gene is going to be transcribed. So if this is a DNA, it will form the RNA. If this is the RNA, this will form the DNA. So in the transcription process, pre-CRISPR RNA is going to be formed. Pre-CRISPR RNA, you can see the repeats forming the hairpin loop-like structure, and this is the viral RNA. Okay, because after transcription, this DNA will form the RNA. So this is the pre-CRISPR RNA. Now, because of this Cas6e and the Cas6f, this pre-CRISPR RNA is going to be converted into the CRISPR RNA. The CRISPR RNA will composed of the hairpin loop-like palindromic repeats, which is you can see here, and this is called the viral genome, which is the viral RNA. Now they will form a complex with the Cas cascades. Next time, when the same virus is attacking, the virus will have the DNA. DNA is complementary to this RNA, so that will bind to this. Now this Cas cascades will activate the Cas3 protein, and this Cas3 protein will cut out this viral DNA. So here, this guide RNA is guiding this Cas protein to chop out or to cut the viral DNA, so that they are preventing the infection of the same virus one more time. So this is the type 1 system. So what you can see here, virus is attacking, the DNA is integrated in the bacterial genome, it is being transcribed, form that CRISPR RNA that will form a complex with the Cas cascades, and then when the same virus will attack the bacteria, then this guide RNA will guide this Cas3 or Cas cascades that will activate the Cas3 and will chop out the DNA. So it is a kind of immune response that bacteria is showing. Type 2 is most common. There are so many modifications have been done till now in type 2. In last 4 to 5 years, most of the scientists are working on this CRISPR so that they can correct the gene. For example, the muscular dystrophy or cystic fibrosis. They are continuously working, but it's not that much easy from going of lab to clinics, but they are working. This is the type 2 CRISPR system. Here the same thing, the virus genome is chopped out, integrated in between the palindromic repeats, and after the transcription, this will form the RNA. So this is the palindromic repeats, the blue, the orange and the red. These are the palindromic repeats, and this is the viral genome. Here there is one more sequence that is complementary to this palindromic repeats, and the whole structure is called the pre-tracer RNA. Now the RNase 3 will work on this pre-tracer RNA and will form the tracer RNA. So the tracer RNA will composed of the palindromic repeats, the viral genetic material, which is now in this case, let's say if it is RNA, and this is the sequence that is complementary to this palindromic repeats. Okay, so this is now the tracer RNA. Now they will form a complex with the Cas9, the same virus. If this same virus is attacking the bacteria one more time, then that DNA will bind to this RNA, which is complementary, and then the Cas9 will chop out the DNA. So this guide RNA is guiding the Cas9. This is the guide RNA. Okay? And this is guiding the Cas9 where to chop out the DNA. So this is a type 2 CRISPR system. Here you can see what is the structure of the Cas9. It's a single subunit, but it contains multiple domains. For example, it contains a REC1 domain that bind to the guide RNA. It contains the PAM interacting domain that bind to the PAM site, and the PAM site is always present in the viral DNA. Here only PAM. Let me show you. In this sequence only the PAM sequence should be there. So what Cas9 will do? Cas9 will interact with the PAM region. Cas9 will interact with the guide. How it will do? Because it has some domains. It has the PAM interacting domain. It has the guide RNA binding domain. It has the nucleus activity domains, which are having the nucleus activity. For example, HNS and the RUBC. So this is the whole structure of this. The function of RAC2 is not known. There is a bridge helix that initiate the cleavage activity. These are the two domains which are responsible for the cleavage activity, or this is nucleus domain that will cut the single-stranded DNA. So how this CRISPR system works? As I said, the gene. Suppose if you have a gene and if you want to target a particular gene, that sequence should be near to the PAM site. And the PAM site, in most of the cases, it is NGG. However, the PAM site depends, the PAM sequence could be different based on the bacterial source from where you are isolating the Cas9. So, but in most of the cases it is NGG, but in some cases it could be the other sequence as well, and we will discuss it. So what will happen? So here you can see this is the guide RNA. This guide RNA is binding to this bacterial DNA, and the Cas9 is recognizing this PAM site. Once there is an interaction between the guide RNA and your target gene, then the Cas9 will recognize this PAM site and will chop this, will cut the DNA, and where it should cut, just the 3 base pairs upstream from the NGG sequence, from the PAM sequence. PAM is what? Protospacer adjacent motifs. So this is your protospacer, and the motif that is near to this is the protospacer adjacent motif, and it is NGG. So once there is an interaction between your guide RNA which you design against your target organ, once there is an interaction, then the Cas9 will recognize this NGG sequence and will cut the DNA, just the 3 base pair upstream of this NGG. So NGG is very important. If you don't find NGG, Cas9 will not work. Okay. So you have to, when you are designing your guide RNA, however, your guide RNA will not contain any protospacer adjacent motif for the PAM sequence, but your target gene, where you want to, what is the gene of interest? What is your gene of interest? That gene of interest should contain the NGG sequence. Okay, and then you have to design the protospacer, and then we are going to discuss it here. For example, this is the gene sequence, and if you think you want to modify this gene, and we found this sequence, let's say NGG, which is N can be any nucleotide. So let's say if it is NGG, this is a protospacer adjacent motif. What you will do, just take the 20, just count the 20 base pairs upstream of this PAM sequence, and that is going to be your protospacer. And the protospacer will be always double stranded. One protospacer will bind to this sequence, other should bind to this sequence, and that you can do by using many softwares. There are many softwares which design the guide RNA, because you have to keep in consideration that the off-target effect should be less. Okay. So what happened in this case? Let's say you design the sequence GAC. So where it should bind? It will bind to this portion. Okay. And since this is a 3' to 5', so you can order just like that. But this sequence will not bind to this. So you have to design another sequence that will bind to this sequence. Okay. And you can say, let's say if it is 5. Okay. So this is, let's say, 5', and this is, let's say, 3', and you are designing the sequence, let's say C, T, T, G, G, C, C, then A, then C, then G, then G, then A, then T, T, C, then T, C, and then it is going to be C. This is now going to be 3 prime to 5. Okay. When you are ordering your sequence, it should be C, C, T, T. So like this, you have to design your experiments or design your guide RNA. You can do it by using the software. Okay. But what if you're not? This is your target gene, you will never get in any. If you are going to NCBI or NSEML, you will not find a double stranded sequence. You will find a single strand sequence, which is the coding strand. So let's say in your coding strand, you are not getting this NGG, but you are getting this CCN, because if it is NGG, the complementary sequence is CCN. So if you are reading from 5 prime to 3 prime, it is CCN. So if it is CCN, you need not to count upstream sequences, you need to count the downstream sequences, 20 base pairs. This, for example, if you design the sequence CAAG, that will bind to this sequence, but when you are ordering this sequence, you have to revert it, because when you are ordering the primers, they should always be in the 5 prime to the 3 prime direction. So what you will order? G, C, G, C. You will not order the C, A, C, G. You keep in mind. You will order G, C, G, C, C, A, C. Okay. For example, in this you can see there is a gene ANGPTL3 and there is a mutation, you can see TCC, and we want to modify this. But we are not getting near to this sequence, we are not getting any NGG sequence, so we are getting CCN sequence. What we will do, we will just count the 20 base pairs downstream from this sequence, and then you will order like we order here. Similarly, you will design your, so this is your PPAM sequence, this is your protospacer. Okay. So the one thing you need to understand that this sequence will bind to this, but what about the other strand? So for the other strand also you have to design your primers. Once you design your primers, you have to add the G nucleotide at the 5 prime position of the forward primer, and in the reverse primer at the 3 prime position you should add the C portion. And why we are adding this? Because in most of the vector, there the promoter is U6, and the activity of the U6 is more in the presence of G nucleotide. At the same time, you will add the CACC at the 5 prime position and CAAA at the 5 prime position of the reverse primer. So your sequence, when you are, and why we are doing this, I will just tell you, but for now you have to understand. So let's say if you design your primers, just add in your forward primer CACCG, and in your reverse primer just add triple AC. And here you can see this is the forward, this is the reverse primer, but both are in the 5 prime to 3 prime direction. That once you are ordering this, you have to keep this thing in mind, and the C will be at the 3 prime position. And why? Because the complementary sequence of the G is C. If you will not add G, then they will not LE. And why we are adding CACC and AAAC? For example, this is the U6 promoter. This is the stuffer fragment which we want to replace it with the guide RNA. So when we are doing the restriction digestion with the BSMV1, we will get something like this: GTGG in the lower strand and GTTT in the upper strand. We have to add in our forward primer CACC. So C will bind to G, A will bind to T, double C will bind to double G. And here this triple AC, triple A will bind here and C will bind here. Okay. What happened in the type 3? Type 3 is not very common, not at all used. The only thing is because in this, the PAM is not, the palindromic repeats are not forming any hairpin loop like structure. There is no tracer RNA. And Cas9 or the Cas cascades can recognize any sequence of the gene. It is not very specific that it need to be near to the NGG. For example, if you see here, it need to be here, the sequential. If this is your gene of interest, if this is the target sequence, that should be near to the NGG. But in this case, type 3, it is not necessary. And once it is done, the Cas9 will cut the DNA. So in the non-homologous end joining, there is a cut of DNA and then there is the ingestion and the deletion of genes, the sequences, and because of this frame shift, what happened? The function of that particular gene is going to be disrupted. This is the knockout. In the case of knocking, you need to provide the donor DNA, and in between this donor DNA sequence, you will insert the sequence that you want to insert in the target gene. So this donor DNA will bind here and will integrate this new DNA in this target gene. This is the homologous dependent recombination, when the recombination will take place. So one more thing I want to tell you, let's say TCC is the sequence which is having the mutation and we want to do the knock-in, we want to correct that gene. And how we can do the TCC, we want to convert this into the TCG. Okay, but you can see here, this is the CCN, which is the protospatial adjacent model for the PAM, and this is the TCC where we want to correct the gene. So if we are converting this TCC to TCG, what will happen? The CCN will convert into CGN, and once the CCN will convert into CGN, the Cas9 will not recognize this sequence one more time. Okay. To prevent the weak labels in the knock-in reaction, we sometimes use this kind of strategy we use so that the PAM sequence is itself cleaved, or the sequence is not anymore there in your sequence. So the Cas9 will not recognize. CCN is recognized by the Cas9 now because it is converted into the CGN. Cas9 will not recognize the new sequence. Okay. But one thing you need to just remember, when you are performing this experiment, it is not at all necessary that every time you are doing the experiment, it is going to be converted to TCC will convert into TCG, or every time you are performing, it is not necessary that 100% of your knock-in is going to be there. There are chances most of the time there may be knockout mutation, and in some clones you will get the knock-in, and therefore the technique is taking time in correcting certain diseases. It's not like that because we introduce the CRISPR and the disease is going to be corrected. There are lots of loops and faults when you are performing the experiment. When you will perform this experiment, let's say, then you will get that not all of your clones will be having the desired mutation. There might be indels as well, so you have to do the experiment multiple times. So as I said, TCG. So this CCN is converted CGN, and there is no PAM now. Okay. So this is a flow chart how we are going to perform this experiment. We digest the vector. We purchase this vector from the vendor that is having the Cas9 and a stuffer fragment that can be replaced by our guide RNA. Then we dephosphorylate this so that they cannot re-anneal. And vector can be binary also. You can use two vectors, one is expressing Cas9, one is expressing just the stuffer fragment that we can replace it by the guide RNA, or you can just purchase a single unit which is containing the Cas9 and the stuffer fragment that can be replaced by the guide RNA. So that's up to you. Once you dephosphorylate, because we want to prevent the self-ligation, and you design your oligos that I told you before, anneal them and then ligate these oligos and these vectors by using the ligation reaction. Okay, do the transformation, select those colonies which are expressing SGRNA, and how you can do that. Vendors usually will tell you if there is ingestion of the SGRNA, and there may be if you will run this DNA, you will get just one band or two band. If there is no ingestion, you will get multiple bands. So like that, you have to select those colonies. And once you have done it, do the mini prep and the maxi prep, and isolate that plasmid, do the transfection and do the selection of those. So here, for the freshers, I just want to tell you, Once you are inserting that vector in the gRNA, vector has a stuffer fragment, and let's say that a stuffer fragment has multiple restriction endonuclease. If there is no integration of this ligation of this vector in the sgRNA, and then because this vector DNA contains multiple restriction sites, so if you are adding the restriction enzyme, that will cut the DNA, multiple bands you will get. Once there is an ingestion, you will get a single band. Like that you can select it. This is just a one way. Depending on the vendor, you can just see how to select it. And once you do the transfection, you will get a number of colonies, and then in those number of colonies you have to do the serial dilution in a 96 well plate, so that your last well contains just one cell. Let them grow. If this is the mammalian cell, it will take many days to grow, 1 or 2 months to grow sometimes. So if you are doing a workshop, it's not feasible to do the experiment in the mammalian cells. It's better to do it in the bacterial cells. So once you have the single cell and the single cell is formed the colonies, then you isolate the DNA from those colonies, amplify the DNA with your primers, that is against your target gene, denature and renature. And then there is an enzyme that will cleave only if there is a heteroduplex DNA. Heteroduplex DNA means the one strand is wild type and the one strand is mutated. Here this is mutated, this is wild. So this reduction enzyme will cleave this, this and this, but this will not cleave this, and this. These three are wild type and these three is both the strands are mutated. So it will only cleave this, and this, and then you will get you have to run this gene and you will get a multiple fragments, and you can see that uh, your gene has been modified. But what if your both the strands have been mutated? You have to confirm that as well. What you will do, you will just amplify the PCR products, denature, renature, and then add the control sgRNA, which is having the wild type sequence. Once you will do the denaturation and when you will do the re-annealing, then you will get something like this. So in one reaction, both of your strands are mutated, you will get single band, because this enzyme will not cleave this, you will get single band. But once you will have add this sgRNA, since it is forming the heteroduplex DNA, means one is the mutated, one is the wild type, because of this control sgRNA, you will get bands. So you can say my both the strands have been mutated. There are many softwares by which you can design the gRNA. The chop is very good, in my opinion, but you can use others as well. This is also good, the Broad Institute one. Like as I said before, the protospacer adjacent motifs, in most of the cases it is NGG. If they are isolated from the pyrogens, the protospacer length is 20 base pair. However, if you see in the aureus case, Saccharomyces aureus, then in that case the PAM sequence is NNGRR, N is any nucleotide, R is arginine and the guanine. Okay, only the purines. And the protospacer is 21 base pair in length. Again, the rules are same. Cas9 will cut the DNA 3 base pair upstream of the this protospacer adjacent motifs. Okay, if it is NGG upstream, it is CCN downstream. So there are lots of mutations has been done to increase its specificity and lower its off-target activity. Similarly, one SC-Cas9 is designed that is 1 kb smaller than SP-Cas9. And why it is done? Because it can be packaged effectively into adeno-associated viruses. And why AAV viruses? Because AAV viruses do not integrate with your genome. The chances of going the same mutation to the next generation is less, and therefore this SC-Cas9 has been designed, which is 1 kb smaller than SP-Cas9, but the function is there, it's not abrupted. Applications, as you can see, wherever you want to modify the gene or correct the gene or delete the gene, then you can use the CRISPR. It is used in the microbiology, biomedicine, and agriculture. Here you can see in 2016 article has been published in which the FGF5 gene is being disrupted, the knockout, the CRISPR, and this particular code is having the more secondary and folic acid along the fibers. And this is the just to reduce the off-target effects. What you can do, you can design the two gRNA molecule directly near to each other. So the chances of off-target effects will be reduced. Agriculture to improve the crop variety, to remove the unwanted species like herbicide resistance weeds or insect pest. You can use it in the agriculture field. My next lecture, I'm going to discuss about this is just the basic, but in next lecture we will just discuss about the practical aspect, and also if you want to perform the workshop, how you can do that. I'm going to describe it in my next lecture so that you can even organize the workshop.